Hello everyone, thank you for tuning in on our channel with a new video. This new video will be about enteric syndromes. This is a theoretical overview and the first part in our series on enteric infections. In this video, we will talk about the theoretical basis, etiology, epidemiology, and pathogenesis, and a little bit of diagnostics and clinic, just enough to understand and grasp the theoretical knowledge needed before we dive into the clinic treatment and different syndromes that will need extensive explaining in each video. Um, the mind map is made using application called XMind and can be found on our Patreon in the link below. If you want to skip to any specific part of the video, you can find it in the description. At the end, there will be extra information about importance of each etiological factor, how to try and find which one is without diving into the clinic too much, and what makes each one specific. And we will go into why each of these agents is really important for us and to know, like the importance of such a theme in infectious disease. So, our overview starts first of all with the etiology. The etiology of enteric syndromes is usually by an infection with viruses, bacteria, protozoa, or parasites, or any of their toxins or products. Usually, they are transmitted through contaminated food or water, and rarely, and in some occasions, they can be transmitted by contact. This is especially if they have low infectious doses. The etiological agents with low infectious doses can cause outbreaks in local international areas and has significance among leading causes of childhood morbidity and mortality. This is important if we have situation with low income country or inadequate organization of healthcare facilities or remote living conditions. That's why gastrointestinal symptoms are still one of the leading causes of death for children in undeveloped countries. So, the pathogenesis of this agent, usually by GI tract, is because the infective agent infects the mucosal surface, which give us some specific symptoms on the mucosal surface. They can have direct microbial invasion of the gut or the tissues under the gut, and these will have their own clinic. These microbial toxins can have effect on the internal structures of the cells or on the tissues or even on the enteric nervous system, which can give us the clinic of this disease. The clinic will be discussed a bit later. The systemic symptoms are usually a result of microbial dissemination via the bloodstream or dissemination of the toxin produced in the bloodstream and its effect on the whole body, or because of host inflammatory response to the infection. This triad will give us the systemic symptoms, and the first triad will give us the GI symptoms. So the first triad is infection of the surface, then invasion, then toxin effect. And in the systemic, we have dissemination, toxin effect, and inflammatory response. These two triads will give us the clinic. Uh, in short, because the clinic will be discussed in its own video, most of them are due to common bacterial, viral causes and are self-limited, and the symptoms resolve within 7 days in normal, non-immunocompromised host. Most of the time, the clinic will not need any microbiological diagnosis because most of the agents have the same symptoms, have the same outcome. So unless we have a severe disease or an outbreak, or we have specific need for screening of the patients, no diagnostic measures are taken. On the horizon of such infections, we can see an emergence of multi-drug resistant enteric pathogens. This will be a problem soon within our practice because non-typhoidal salmonella, shigella, and vibrio cholera, they have increased frequency now, especially in places where we will have disruption of normal sanitation or normal civilized activities in cases of earthquakes or humanitarian crisis. And within the nosocomial world and the developed world, we have Clostridioides difficile infection, which is becoming more and more resistant to different types of antibiotics. So our work with gastrointestinal infections is not limited and is not done with all these developments happening all the time. So how to classify these? There used to be an old classification system that's now only used for general diarrheas, and now we have a different classification. Gastroenteritis, which is one of the main symptoms of the main disease of gastrointestinal infections, has two main syndromes, which is diarrhea and vomiting. These are the main GI affected parts. They can come alone or both, with or without systemic symptoms. Diarrhea is the main syndrome where the symptom is diarrhea, dehydration, and systemic effects of intoxication or dehydration. It is three or more loose stools in 24-hour periods. It is acute if less than 14 days and persistent if more than 14 days. After 14 days, we will have a specific algorithm of treatment, which will need more diagnostic search and more aggressive treatment. And we can also classify it into community acquired and nosocomial. It is important because the agents that come within nosocomial, like Clostridium difficile, Acinetobacter, Pseudomonas, are a little bit different than community acquired, which are mostly Campylobacter, Staphylococcus, staphylococcal infections, Salmonella infection, and E. coli infection. So the first thing to do when we have a patient with gastrointestinal infection is to know which of the two or both of them syndromes is he having. Is he having systemic clinic? Is he having any complication? How much is a diarrhea to classify the diarrhea? If it's acute or persistent, and we should know if it's community acquired or nosocomial, and to know his immune status. These are important to classify our patient to know later what to do with them. And now we go to the epidemiology. The first important thing to know about it is risk factor. And once again, by knowing the risk factors, we can know how to deal with our patient depending on his risk category. So, we start first of all by undernutrition. Undernutrition is the biggest risk factor, especially in children and especially in micronutrient deficiency for complications. Diarrhea in such children will exasperate itself and nutritional deficiencies, and it becomes like a closed circle. When the patient has low nutritional status, he gets diarrhea, he loses more nutrients, and it continues. Kids with this syndrome will have more complications, more severity, and higher morbidity, and will have longer hospitalization than um, other children. And this is mostly important in children. Immunodeficiency We'll not go more into that, but people with immunodeficient status will have a little bit of different and more severe and more resistant etiological factors. And these patients will have longer stay in hospital, will need more care, have more burden on the healthcare system, and have higher mortality. Another risk factor is the time of the year. We have seasonal pattern in different climates. In temperate climate, we can have winter vomiting disease, where we have viral infections going through when people are living in small quarters, especially in cold countries when people will be packed into uh, heated areas. And in tropical climates or moderate climates, we will have distinct summer or late spring peaks in GI illnesses. Another important risk factor is age. The first age group to check for is children younger than 5 years. They have the greatest morbidity mortality from GI infection. Why is this important, and what are the risk factors within age? The first mechanism is when these children start to lose the maternal antibodies. These antibodies are important to control the infection until the baby's body start to produce its own antibodies. If we have discontinuation of breastfeeding, it is an important reason because the mechanism where milk glycans can have soluble receptors that inhibit pathogens. This inhibition will stop the pathogens from adhering into the target receptor on intestinal mucosa. And adhering is the first step in all GI infections. By stopping it, we are slowing down the infection spread and giving the baby time to react by itself. Breast milk contains high level of secretory IgA, and if we know from immunopathology, we can know that IgA is the specific immunoglobulin for epithelial surfaces. And this is a crucial role in mucosal immunity and can help the patient fend off adherence and invasion of these bacteria or viruses within his gut. By stopping breastfeeding, he will lose the IgA and the glycans. And breastfeeding environmentally reduces the risk of exposure to contaminated foods since the baby is getting fresh milk from his mother. This will stop any contamination by storage or by abnormal administration of food to the baby. That's why the WHO recommends exclusive breastfeeding for the first 6 months of life. Another risk factor for babies younger than 5 years old is when we have high rate of antibiotics within the first 2 years of life. This can predispose our patients to serious disruption in his microbiome and his bacteria within the gut, the protective bacteria, and predisposes him to more severe infections. And these children have higher rates of rotavirus, enterotoxic E. coli, uh, enteropathogenic E. coli, Shigella toxin E. coli, which produces Shigella-like toxin, Shigella by itself, Salmonella, and Campylobacter, and other uh, typical agents. These are the agents that usually come with children younger than 5 years old. The other individuals that are also susceptible to complications of enteric infection are people with who are age more than 65 years old, and these have the same risks as young people, but their complications are usually more into dehydration and the compensation of other comorbidities. Another risk factor to know is gastric acidity. Gastric acid is considered to be one of the first lines of defense for bacteria coming into the GI tract. A lot of them are really susceptible to high acidity and will be degraded. And the infectious dose that we will, like if we have 1 million bacterial units coming from our mouth, by getting into the intestine, it will be reduced by the acid into, for example, 1000, which is less than the infectious dose for most of the intestinal bacteria. When the patient is having low acid secretion for some reason, it might be extensive use of PPIs, or it might be some genetic predispositions. These impaired acid secretion will increase the frequency and the severity of infection. Within the same reason, another protective factor is intestinal motility. The motility of intestine will give the bacteria less time to aggregate and to adhere into the mucosal surface and will lead to toxins and produces of this inflammation going faster out of the body. That's why normal peristaltiasis helps maintain the enteric microbiota and clear pathogenic bacteria. When we have low intestinal motility, we will have extended infection. And if we have really high motility, we will have micronutrition problems and we will be having diarrhea, which is not related to our infection. Another protective factor is the intestinal microbiome. Our microbiome is usually consisted by a lot of bacteria, such as bifidobacter, such as enterogenic bacteria. These bacteria will have a competition with other organisms for nutrition and specific niches within the intestine, like crypts and intestinal binding sites. This will prevent adherence and colonization of infection within the GI tract. And these bacteria will have will kind of maintain the low luminal pH, which is another protective factor. And the compounds produced by these protective intestinal microbiome will inhibit the pathogens. The factors that can disrupt our intestinal microbiome is prior antimicrobial exposure. Other risk factors that can be dived into, but I don't think that it's clinically relevant for us. It's genetic determinants and nutritional status. It's like patients who have genetic disturbances that can influence any of the other risk factors, or nutritional status, where the patient has specific nutritional deficiencies that have their specific clinic. Now, within the epidemiology, we will go into the occurrence. Within occurrence, we'll understand why it is important for us to learn and understand intestinal infections. First of all, the outbreaks of foodborne gastroenteritis is common even in high-income countries. For example, in America, in the United States of America, we estimate that there is 9.4 million disease every year, with almost 60,000 hospitalizations and 1,351 deaths. This was, I think, in 2019. The statistics are from. Um, the etiology of occurrences with outbreaks. We're not talking about community acquired only, we're talking about big outbreaks. Was Campylobacter during the 2000 Toxoplasma, which had another outbreak within the US. Listeria had another outbreak. Salmonella enterica, which is quite common. And in 2011, there was a multinational outbreak of Shigella toxin, Shigella-like toxin enterocolitis. And during this outbreak, we had a lot of hemolytic uremic syndrome cases. We will dive more into that when we will have the video specially made for E. coli. Another important thing to know in occurrence is the persistence of nosocomial infections, GI infections. This is predominantly because of the Clostridium difficile, which is now Clostridioid difficile within the new classification. The spores can contaminate the hostile environment and the hands of healthcare workers. They can live for up to 6 months on surfaces, and are resistant to alcohol-based disinfectants. The spores are resistant to gastric acid and then can spread from one patient to another, even from the hands of an already discharged, from one discharged patient to another by the hands of the healthcare worker. This is important. This is why it's important to use proper sanitation when dealing with GI patients, especially with patients who are on high doses of antibiotics, since antibiotic use will kind of inhibit our microbiome and will lead to aggregation of Clostridium difficile. And this will also be discussed in depth later. Other important pathogens to look out for are Klebsiella oxytoca, toxin-producing Clostridium perfringens, and we have common norovirus healthcare setting outbreaks, but usually they have self-limited outcomes, unlike Clostridioid difficile, which has lethal outcomes usually. Now we will go into microbial factors. These are the factors that the bacteria or the agent can have by itself, which will lead to talk about bacteria in here, since most of them are bacterial. The ones that have the most complications are bacterial, and they are the most understood. So, in bacterium, the genes required for colonization and infection are carried on laterally, which means that these elements can get transferred from one bacteria to another in specific islands of pathogenicity. These virulence plasmids, transposons, and bacteriophages are usually transmitted and carried laterally on these bacteria. That's why these pathogenic bacteria can produce and secrete number of proteins which can modulate the interaction with the host. These proteins, we will talk about them later in the steps. Gram-negative bacteria have within their walls specific proteins that can be secreted in the inner and outer membranes, and even a subset of proteins that can be excreted out of the cell. The mechanism of survival and aggregation of these bacteria within our body, first of all, are nutrient acquisition. They can take the nutrients produced by our body and use them for themselves. They can produce proteases and cleave the human IgA1, which will stop their inhibition. They can be motile and have chemotaxis toward intestinal epithelial surfaces. That's why they can move and find the places which are more probable for survivability of this agent. And they are capable of sensing the environmental signals that indicate that they are within the human GI tract. That's why the behavior of these bacteria and the secretions of these bacteria will differ from when they are before and after, because when they are triggered, they will coordinate a regulation of virulence genes, which will cause the infection. These virulence genes are the genes that are responsible for the steps that can be used to produce the infection and the clinic. All our infections start the same way, but some of them stop on this step and some of them continue. All of our etiological factors will need adherence and attachment to the GI mucosa, either be it a bacteria, a virus, a parasite, or even a toxin of a bacteria. They will all be attached to the GI mucosa. The attachment has its own receptors, and each receptor is specific for each etiological factor, and it can sometimes be helpful in diagnosis. Some of these factors will go into cellular invasion, where will not only attach into the cell, but they will invade inside the cell and into the layers under the cell. Some of them might even produce exotoxins that are proteins that go outside of the bacteria and can interact with our body and our GI tract. These toxins, whether are exo or endotoxins, they change the epithelial cell physiology. And another step that can be used is the loss of brush border of the digestive enzymes, and they can cause cell death. All of these steps will lead into increased motility. We will have more fluid secretion and less fluid absorption because of the loss of brush border. We will have inflammatory cell influx and inflammatory reaction, and can even go as deep as intestinal hemorrhages. An important part of our factors for infection are the infectious doses. The inoculation of microorganism needed to produce infection can vary across organisms between different hosts. Some bacteria need 1,000 copies to give an infection, and some of them need few million. So to check this, the scientific society has decided that the median infective dose is the best unit of measurement, and this represents the median amount of units needed to infect 50% of the population. So, for example, if the ID50 of an infection is around 1000, it means that if we give 100 people 1000 units each of this bacteria, at least 50 of them will get the infection. And as we said, the ID50 is influenced by gastric acidity because ID50 is the amount of bacteria inoculated by while taking food or within the mouth, but it doesn't tell us how many of these bacteria will actually make it into the gut. That's why the ID50 will change depending on the gastric acidity, which will lower the amount of this dose coming into contact with the intestinal mucosa. So examples of infective dose. Very low infective dose, median infective dose, is for bacteria that are resistant to gastric acid. For example, these are E. coli and Shigella, cysts of Cryptosporidium and Giardiasis, norovirus, and Enterobia histolytica. These can have as low as 10 units or 100 organisms. This is a really low infective dose. And these etiological agents have high probability of person-to-person spread and can influence the epidemiology since they can cause outbreaks. For example, the norovirus outbreak that can come within specific healthcare facilities. Most agents that infect the IG tract have a moderate to high infective median dose, which is around 10 to the power of 3 or 10 to the power of 8, which is 100 to 100 million, no, 1000 to 100 million units. And a higher ID50, such as 10 to the power of 10, is seen in Yerseniasis. But as we said, most of them have 10 to the power of 8. Now, examining each of these mechanisms of action and factors of our bacteria, we can start, or other etiological factors, we can start by adherence and attachment. In this case, we will talk about bacteria because if we go into the viruses, each of them has its own mechanism. So the interaction of bacterial surfaces protein with the host cell surface proteins is specific to each species, but most of them has pili or fimbri, one or both, and they are the most common mechanism used. As we can see, the pili are long appendages made of polymers of a protein called pilin, and the fimbri are almost the same but lower. They have other different functions within the life cycle of the bacteria, but within the scope of our clinical work, it's not important for us. All that we know that they are both pro-inflammatory and pro-adhesion. So other proteins may coat the tip of these structures, and these proteins can help with further adhesion. For example, we will have the bundle-forming pilus in enteropathogenics um, E. coli, which will help in the pathogenic part of this type of E. coli. We can have a colonization factor antigen that's also on the fimbria and pili of ETEC, which is enterotoxic enterocoli. And this will help these bacteria colonize our GI tract. And this is an important factor within the hemolytic uremic syndrome later. The aggregative adherence of enteroaggregative E. coli is also a specific protein that will help different bacteria aggregate to each other and give some kind of biofilm in the enteroaggregative E. coli. Some bacteria will have non-fimbrial adhesions on their outer membrane, and these are called fibronectin and will help them bind into the epithelial surfaces. One important type of attachment and adherent protein is the toxin correlated pilus TPA, TCPA of Vibrio cholera. This is helpful to make the toxin and the bacteria create microcolonies on the GI surface rather than binding to specific intestinal receptors. This will help them make these microcolonies, and then the microcolony by itself will have specific attachment into the wall of the GI, where the bacteria itself doesn't have any specific receptors for the GI tract. And the next part of this mechanism is invasion. Certain bacteria, especially the enteropathogenic bacteria, will invade into the cell or through them to the underlying space. This will induce a lot of pro-inflammatory cytokines like EL8 and others, and will mediate the influx of inflammatory cells. This kind of process will give us a more inflammatory type of diarrhea or infection, and it's usually part of a different kind of diarrhea. We'll take a small break now to go into the different type of diarrheas, where we will have by another classification, which is non-infectious classification, different type of diarrheas. We will have osmotic, exudative, secretory, and motor. In osmotic, we have specific molecules that will take out water from our bloodstream and put it inside the GI tract and will transfer it to make us diarrhea. Usually, this is for lactose intolerant patients and is rarely infectious. Exudative is, for example, within cholera and toxin mediated. It's when the toxin or the bacteria stops at the GI surface and will increase the exudation or the secretion of water. Um, I'm sorry, I kind of mixed them up. This is the secretory. The secretory will be only in the first surface and will increase the secretion of the water. That's why it's called secretory. Exudative is when we have inflammation, and this what happens when we will have invasion. We'll have increased intestinal contents, we will have mucus, we might have blood, we might have even pus inside our watery diarrhea. While in secretory, it will be only watery diarrhea. That's why exudative diarrhea will happen when we have an invasion, and secretory will happen only when we have attachment or toxin mediated type of diarrhea. Motor can happen within any of those three. The motor factor is usually not a specific type of diarrhea by itself, but just an extra factor in each one. So we go back into the invasion part, and as we saw, this pathogenic process is important because it will give us a different type of diarrhea, therefore a different type of pathogenesis, then a different type of clinic. And sometimes it prompts different type of treatment. The last microbial factor to go into is the toxin. So most pathogenic bacteria produce a protein called exoprotein, which is a class of protein, not a protein by itself. And several of them will have A part or subunit and B subunit. The A usually is enzymatically active and will change the cell physiology of our GI tract. And B is the B binding subunit. That's why A active, B binding. The B part will have specific receptors on our cells or eukaryotic cells, which can be any of our GI tract cells. It can be the secretory cells, it can be the epithelial cells, it can be the pro-inflammatory cells. It can have receptor on any or each of them. Then the E part are internalized, they catalyze specific activities within the cell and they lead into alteration of cell physiology. We will go a little bit deeper into this. First of all, by explaining that we have three types of toxins. We have neurotoxins, enterotoxins, and cytotoxins. Neurotoxins are the ones that we should understand because usually neurotoxins come by themselves without the bacteria. We can ingest neurotoxins preformed. For example, one of the most classic cases, we have a bowl of rice in our fridge. We have bacteria like Bacillus cereus or Clostridium perfringens, and they will produce their own preformed toxin within the rice. We ingest the rice, the toxin goes into our body, and this toxin will have binding into our cells, into our GI cells, will change their physiology and will lead into having diarrhea and cramps. And this is why when we have specific diarrhea, it is recommended to avoid antiemetics or motility stoppers or antidiarrheal drugs because they can let these enterotoxins stay longer in our GI tract and they can increase the time and the duration and even the complications of our disease. That's why when we have ingested neurotoxins, it is better not to treat them with antidiarrheal. The most enterotoxins come from Staphylococcus aureus, which has one of the most potent. It acts on the central autonomic nervous system, will have acute upper GI syndrome, and especially vomiting. And a similar neurotoxin is produced by Bacillus cereus. Usually, it's within 1 to 6 hours of ingestion, and it has preformed toxin within the food by itself. And as we said, a classical story is the rice in the fridge. It can also produce enterotoxins, like each of those bacteria can produce at the same time neurotoxin and enterotoxin. That's why it can produce in the beginning vomiting and then diarrhea after 8 to 16 hours. So, since we have no specific diagnostic factor for this or test, usually this will be clinically diagnosed. Another neurotoxin that doesn't have a GI activity is the botulinum toxin. It's produced by Clostridium botulinum, or other clostridial disease or species. Usually it's within not well prepared conserved food, and this toxin will give us flaccid paralysis of multiple parts of the body, and it's quite dangerous systemic disease that we should look out for. Now we go into enterotoxins. A number of pathogenic bacteria produce enterotoxins. They act directly on the GI mucosal cells and stimulate net fluid secretion. That's the important pathogenic part. For example, we have the cholera toxin. It is an A1B5 protein. It is exported out of the bacterial cell. The B part will bind into enterocyte. The A subunit will go into the cell. The protease will reduce it and it will enter the cytoplasm in a reduced form. It will lead to increase in CAMP, and this increased CAMP will increase net fluid secretion through the apical chloride channel into the GI tract, and we will have diarrhea. That's why, for example, within cholera infection, we rarely see any temperature because we have only enterotoxin. We don't have systemic disease formulating. Another enterotoxin, which is ETEC, it produces a heat labile enterotoxin. Usually it's written HL toxin. It is very similar to the cholera toxin in protein sequence and have the same action. So it will bind and it will activate the intestinal gluconate cyclase, which is similar to the CAMP cycle within the cell. Now, if we go into different type of toxins, we can go into the cytotoxins. This is more in the dysentery type of infection, of intestinal infections. And this have more destructive pattern, will have more inflammation and more systemic uh, type of disease. So the Shigatoxin family is produced by the Shigella dysentery and STEC. The B part will bind into the receptor called GB3. It is internalized. It goes retrograde transport from the endoplasmic reticulum into the nuclear membrane. The A subunit will have the same nicking, reducing, and entering into the cytoplasm, where it will have specific adenine residue inhibiting protein synthesis, and it will cause cell death. Because we will have this residue inhibiting protein, it will be inhibited, and this cell death will increase our entry of these bacteria into the bloodstream because we will have disruption within the cell wall. We will have increased inflammation, we will have increased diarrhea, we will have increased intoxication. And this toxin that is rich with the GB3 receptor can cause vascular damage in all endothelial cells. It will cause bloody diarrhea and it can lead to hemolytic uremic syndrome, which will have a video by itself. C. jejuni and some strains of E. coli produce a cytolethal distending toxin. This will produce cell cycle arrest and will lead to cytoplasmic distension and cell death. This will have a similar clinic. Actually, most of them will have the similar clinic. Another cytotoxin to look for is the cytogenic strain of Clostridoid difficile. And it has two protein exotoxin, toxin A and toxin B. After binding, the toxin are internalized, and we will have disaggregation of polymerized actin, We will have opening of tight junction, cell rounding, and subsequent cell death. This is why we will have pseudomembranous colitis by a specific kind of pathogenesis. Here we can see it where the extracellular pathogen will come. Usually in this case, we will talk about Clostridioid difficile. It will go inside the cell. The aquaporins will open. We will have accumulation of calcium within the cell. The tight junction and the specific skeletal system of the cell will be disrupted. Water will be start to seep through these tight junctions. We will have increased production. And if we are talking about clostridial, we will have detachment of these cells. That's why we'll have pseudomembranous colitis. If we are talking about Shigella and Salmonella, we will only have the destruction of tight cells and increased secretion and diarrhea with increased motility because of inflammation. And this is especially in cytotoxins. If you go down to check other types of infections, we can check, for example, the neurotoxin, where we can see, the enterotoxin. Enterotoxin will bind, will go inside. We have accumulation of calcium, accumulation of CAMP. This exotoxin can go inside the enteric system, which will increase the motility, will have increase of water production, decrease of reabsorption. Here we have stop of these channels, and it will all go inside the intestine. Kind of the same happens with rotavirus, for example. The virus comes in, will change the physiology, the tight junctions will open, and we will have almost non-inflammatory secretion of water. That's the difference between viral, for example, and toxins, for example. But in both of them, we will have low inflammatory type of diarrhea in compared with the high inflammatory type, which will give us cell death at the end. In this table, we can see different types of toxins, and each toxin produced by which bacteria. For example, Staphylococcus aureus, which toxins it will have, and this will be important later because some of these bacteria can be treated by antibiotics. Last table to go into, it's the table of agents and what is special about each agent. In viruses, for example, we will have adenovirus, which is the leading cause of infantile gastroenteritis. It's infantile and not neonate. In astrovirus, we will have outbreaks. Neuro, it will be in specific environmental and winter vomiting illness and within small outbreaks within specific healthcare facilities or closed facilities. Rotavirus is the most common global cause in young children, but now it's getting lower because of vaccination. If we come into bacteria, the most important is Bacillus cereus, especially in ingestion of specific toxins. E. coli species, the enteroaggregative in young children and malnutrition. Enterohemorrhagic, it's the one that will cause hemolytic uremic, and it's the Shiga-like toxin producing. Enteroinvasive, dysentery. Enteropathogenic, acute watery, and enterotoxigenic is usually with ETEC, and it's usually with travelers. Listeria for uh, pregnancy complication with systemic and with old people. Non-typhal, it's for cattle. Cholera, it's in lack of sanitation and humanitarian crisis, which can be expected now, for example, in Turkey with the earthquake happening. Staphylococcus aureus, It's ingestion of toxin, and cholera can still be taken by ingestion of contaminated seafood. The last one to check is protozoa. Protozoa is mainly for people with opportunistic and HIV and children of low income. We will go into clinic in the next video. Thank you for being with us in this video. The whole mind map can be found on our Patreon. Any donation on the Patreon will be really appreciated. Link will be in the description. And keep in tune for the next video where we will talk about diagnosis, clinic, and how to treat our patient. These videos are not only to study for exams. These videos are more directed to understand what we will do with our patient. That's why we spend so much time, especially on understanding the mechanism and the theoretical overview and basis of everything. We studied and understood all of this even before starting to study the disease itself. This will be important later as everything will come into place like a puzzle. Thank you for being here. Please subscribe and like, and hope everything will be okay with your patients.